Yeah, I can. Okay, go ahead. Hello everyone, and today the group number 6 will present 3 species. But in this case, Guilherme and me will present or would like talk about the Brazilian medicinal plant. In this case, this is jabuticaba. Jabuticaba is known as the Brazilian grape tree uh, because it's very similar a common grape or the grapes. In this case, jabuticaba, its family is Myrtaceae. Its scientific name is Plinia cauliflora, and its popular name or local name is Brazilian grape or jabuticaba or jabuticaba. In this case, a particularity of this species is the flower. For example, we can see the flower and very grow on the main trunk, for example, and branches. The fruit has purple black of the peel is purple black, for example. Guilherme, continue, please.
The fruit is consumed by eating the ripe fruit pulp. It's also used for making and jam and liquor. This has led to an increasing value in the market, forcing resort as well. For example, the fruit peel is used for medicinal purposes with purposes such as antioxidants and diabetics, among others. Such results drive analysis that consequently explore new uses for the fruit. Next, please.
The next slide, please.
Oh, okay. Sorry.
Thank you, Farid. Okay, we can see our medicinal properties in this fruit mainly. Because it has antioxidants power in this fruit mainly. This is powered are related to medicinal activities and health benefits. Okay, for example, biological activities present in the fruit, uh, like anti-inflammatory, hypoglycemic, analgesic, um, reduce obesity, for example. But it's very interesting to find other research talk about the biological potential in the leaves, but it's not there are not reports enough about this part in the plant, or talking about the leaf related biological potential. For example, in these articles talking about the potential in the leaf, again, um, Chagas disease, for example, Chagas disease is related with trypanosoma. It's very important to continue in this area to comprehension about the other potential or biological potential in the leaf in this fruit. About the acrobat plants, there is not information about the sport, and additionally, a commercial occurrence of this fruit are still scarce or is not enough. Thank you. Continue, please.
Next slide, please. Well, does the fruit have sociocultural and environmental challenges? Yes. The literature contains some more information. For example, challenges relate to cultural appropriation, and equitable access to benefits. For example, the environmental challenges in Jabuticaba are mainly related to use loss of the natural habitat of the jabuticabeiras due to the use of the land for other crops of greater economic importance and difficulties with species management and sustainability. Next, please. Um, the transcriptomic data is summarized in the identification of genes related to the biosynthesis of anthocyanins and the regulation of jabuticaba skin pigmentation.
Okay, with related about the metabolomic data, this information it's related with phenolic compounds, mainly. There are many reports about the chemical compounds in different parts of the fruit. For example, in the peel, it's abundant in phenolic compounds, especially anthocyanins. Anthocyanins is related with antioxidants power in the fruit. Other parts, for example, the seeds are rich in hydroxylable tannins, for example, and leaves. There are characterizations, flavonoids class, or other phenolic acids, for example. In the pool, there are reports about sugars contained, especially fructose. Continue here, please.
Genomic data is also scarce. One paper focused on the genomes of the plastid Plinium ciliandra and Plinia cauliflora, within the Myrtaceae family, reveal structural and evolutionary characteristics, highlight their phylogenetic distribution, and proving evidence of protein selection in several genes. Next slide, please. To conclude, think about future perspectives, think about how we could more efficiently explore this natural research through molecular genetic methodologies. I study has also considered this, and we corroborate. The first step would be the creation of genomic research for the Myrtaceae family, including long-reading sequences and characterizations of plastid genomes. It's a crucial step. With this, we will be able to fill gaps and knowledges and facilitate conservation and exploration efforts for this species. That is all. Thank you.
Okay. About the Mortinho, like is our food species, Vaccinium floribundo, better known as Mortinho, is a wild shrub endemic to Ecuador and Colombia. This white fruit is similar to a blueberry and is destroyed by the Andean paramos and humid mountain forests. And Mortinho requires specific climatic characteristics for its development. In Ecuador, we consider it as a ceremonial fruit because despite being used in different preparation, it's generally used for the preparation of a traditional drink for the Day of the Dead, known as colada morada. This fruit has a high potential because it has a high antioxidant content. Some challenge about environment or sociocultural is until now Mortinho has not been domesticated, and the obtained of this fruit is limited to harvest in productive months. In addition, population have faced setbacks due to the deforestation, alteration in land use, habitat fragmentation, and disturbance resulting from their extraction. Next slide, please. Okay, about the resources that I found. Sincerely, there are not much genomic, transcriptomic, and metabolomic resources for this spice. But however, I found the following about transcriptomic data. I found one publication in 2021. They comparing the expression level between genes, many enzymes, structural genes, and regulatory elements have been observed as transcription factors involved in anthocyanin biosynthesis. As genomic data I found the first complete Mortinho chloroplast genome sequence in 2023. This study is one of my favorites because it was conducted at my university, and as metabolomic data, what I found was about the components that Mortinho contains and are a potential functional food. Also about the use of Mortinho as a natural colorant. Next slide, please. Okay. Next. Okay, what approaches I want to take? First, it seems like a good option to carry out a study similar to this one, where they also use blueberries, and especially because of the antioxidants it has, and they could be a good option for the study. Here, the antioxidant here they do the antioxidants capacities of 15 commercial raspberry grown in the northern of China were evaluated, and their anthocyanin profiles were determined by LCESIMS. The higher phytochemical contents and the antioxidants activities of raspberry varieties, meaning that their consumption will be more beneficial for health. And I think that is a good idea. Next, please. Okay, another option that seemed interesting to me is the alteration of many metabolomic pathways in plant cells and organs that contribute to the balance of the metabolite profile in the body. Okay, given the enormous diversity of metabolomics compounds that plants produce, the integration of omics data, for example, the integration of genomics and metabolomics, I think is a good idea to make some similar with the Mortinho. And because I think that is interesting to know the system-wide impact that could result for genetic engineering of crops, as well as analyze the response of nutrient stress And finally, I think that a challenge that I can take in count, the story of Mortinho is limited by the difficulty in developing in other types of environments that are not its natural habitat, and also by the indiscriminate abuse of this consumption, taking into account that there are not crops of domestic. Okay.
So it's already 15 minutes. I'll be very quick. So I picked a plant species, which is in legume family, and it's actually wild relative of common bean. And it's one of the reason I chose this because one, it has ecological importance, and second, it has agricultural food related importance. And because it's also wild relative, and we should more focus into crop wild relative because they have higher genetic diversity, so that we can find the interesting good genes and introduce them to other plant species, right? So, and usually their usual habitats is in United States. Mainly, they have two kind of ecotype. One is beach ecotype, and another is inland. And interestingly, the beach ecotype is actually salt tolerant. So, and why I focused on legume species, beans are very famous in both North America and South America. Even had lunch, beans as lunch. So, and they are very high in protein, fibers, So, and very like this species is widely dispersed, and North Carolina, where I did my PhD, is very like you can find them in North Carolina, and using these wild relatives, we can actually improve the common beans, So like how salinity tolerance work, because one of the focus of the sand bean, which is very highly salt tolerant, and there are so many like metabolic pathways, like interconnected pathways that actually responds to these saline tolerant, like, and they actually help plants survive in this adverse environment. So focusing on this quality of this species, uh, like I also looked up what kind of research has been done so far. There only have been one transcriptomic studies for two genotype of this Staphylo or this Strophostyles helbola. Only just this transcriptomic studies I could find. And then there is also a genome sequencing I found, but it was not very advanced. Like it was genotypic based sequencing, GBS. So what approaches we can take to find out more about these interesting species, that we can actually choose different ecotypes of this plant species from variable environments and do genomics and metabolomics. For example, we read about long read sequencing from the Marks et al. Paper, right? So we can apply that with more ecotypes to find out what genetic diversity they have. But one challenge is like funding, right? So it will need collaborative approach, like maybe some collaboration with eastern part of US, or also this plant grows in Canada. So collaboration always helps. And another approach we can do after having this comparative genomics, we can do comparative metabolomics across different ecotypes from various environments to see if genetic diversity in this crop wild relative also causes metabolic diversity. So that's it. And I had a very good time with you all. I hope we'll keep in touch. And we have a now WhatsApp group. Like, I hope we can keep in touch. And I really like this course. We all can actually now unmute ourselves and say thank you. Do you want to do that? All
Thank you,
together.
too. Right.
Unmute yourself, Ivan and Guilherme. Okay, so that's all. I will just stop recording. It's almost 20 minutes. It's okay, I guess. So
Yeah.
let me stop recording.